All right. Hello. Good morning. Good afternoon. Good evening, everyone, wherever you are. My name is Denis Jabaudon. It's a pleasure to welcome you on this second or third edition of the worldwide neuro developmental events. I hope you're doing fine and that things are moving forward the way you like today. So today, our guest is Bassem Hassan from the Paris Brain Institute, Institut du Cerveau in French. And before I introduce Bassem, I'd like to remind you a few things about the way this seminar series is organized for those of you who haven't participated before. So you will see, well, there's this sidebar on the right where you can make comments, any type of constructive comments that you feel like. And also there's this bar at the bottom, this tab, where you can ask a question. So as the presentation moves forward, feel free to ask questions. I'll copy paste them if necessary or comment them, and they'll be ranked based on the type of votes. You can vote on the questions that you would have asked yourself. So feel free to do that. It's meant to be as interactive as possible. Also, I'd like to draw your attention on the fact that there's a new seminar series on neurodevelopmental disorders that will take place on Tuesdays at 5 p.m. They're put together by Gaia Navarino, Laura Cancheda, and Camila Bellone. So yes, if you're interested in development, I guess it's a nice sister series, and do have a look at the excellent array of speakers. Voila. So I'd now like to introduce Bassem before his presentation. So Bassem grew up in Germany and in Lebanon, after which he did his PhD and postdoctoral training at Baylor College of Medicine in USA. And focused on genetic tools to identify different types of circuits in the fly brain. So he actually discovered the dorsal cluster neurons. I didn't know about them, but it is an anatomical entity, which are in the optic lobe of the Drosophila, and which are interesting because at the larval stage, these neurons will extend or not an axon to the opposite side of the brain, and Bassem characterized in particular some of the molecular mechanisms that control this, and made the interesting observation that while some neurons did project, others did not, which I suppose fueled his long-lasting interest in trying to understand how deterministic development of neuronal systems are. After this training and early career in the US, he moved to Belgium, to the VIB in Leuven in 2001, where he stayed until 2016 before joining the Paris Brain Institute, by then Institut Cerveau Moelle Épinière, where he became the director, scientific director in 2019. So as you may have guessed and probably already know, Bassem has this very original line of research aimed at trying to understand the role of deterministic and non-deterministic factors in brain development. And this originality and the high quality of his work has led him to receive a number of awards, including an Einstein Fellowship, Allen Distinguished Investigator Award, and this year, a couple of months ago, the Roger Desperge Prize. Little personal note on Bassem. We were actually chatting before we went live, and I tried to ask speakers for a little personal anecdote, and I'm going to tell you the one I wrote about him, the one he told me. But I realized also we both realized we had shared a near-death experience together, so that'll be for later times. I was the driver. I must admit, I was the driver for this near-death experience. So when we meet in 3D, we'll be both very happy to share this event. I think Bassem's comment, though, is more interesting. And let me read what I wrote. So during his early years as a student in back then war-torn Beirut, Bassem had a passion for literature, in particular for poetry. When the time came for him to make a choice, he rationalized that quote unquote, as a scientist, I could still write poetry, but as a poet, I could not do science. So he chose to decipher the language in which nature writes her poem, and as he put it, There is poetry in biology in both its form and content, and the pursuit of that beauty is both rigorously rational and emotionally uplifting. So Bassem, in these challenging times, I think we can all do with a little bit of emotional uplifting. We're counting on you. Very happy to have you here, and the screen is yours.
Thank you, Denis. Can you hear me? Is this working?
For me, yes. I guess okay. People would have complained if not.
Thank you so much for having me in this series, and what a great idea in this weird time that, you know, getting us together, getting people from all over the world together. And I want to thank the 455 people who are here. Thank you so much for coming to hear me. That's so cool. What I'll do is I'll share with you. So my lab's interested in how the brain.
You want to share your screen already, Bassem?
Yeah, I'll do that in a second.
Okay.
How the brain develops. We're interested in early events like neurogenesis, but we're also interested in the development of neural circuits and how the development of neural circuits impacts behavior. I'll start by sharing the screen here. It's not that. Sorry, 1 second. It's being slow. Okay, it's not letting me do the sharing now. I
can talk about the near-death experience in the meantime.
So I might have to like reload the whole thing because it's just not allowing me to do the screen sharing.
So you don't have the little button on the top?
Yeah, but if I click it, nothing's happening.
You want to refresh like we did last time? You want to click the refresh thing?
That's what I'm going to do. No. I'm going to do that. Sorry, everyone.
Let's see.
Okay, I think I'm back, right?
Yes, you are.
Okay, share screen. Okay.
Yep. All
Is
good.
that working?
Yeah, well done.
Okay.
So I'll zoom in on the screen.
Can you see the presentation now?
Yeah, it's all good, Bassem.
Okay, great. Okay, so all right. So what I want to tell you about is a story of how individuality might emerge during development and how that translates into behavior. So really, this is the problem, right? I mean, we know that the brain is extremely complicated and that connectivity in the brain, the connectivity of neural circuits is incredibly complex, and yet somehow it works, and it works in a fairly robust way. In fact, so robust that it's, at least on average, pretty predictable, right? So this is the conundrum we all want to solve, and if we make things very, very simple and we look at, you know, the simplest behavioral paradigm, which is innate responses to environmental stimuli, then maybe there's a chance we can try to sort out how the complexity of the neural circuits in the brain might actually tell us something about how the brain works. Now, we'll start from the basic assumption that brain development is important for brain function. And of course, this is true in a trivial sense. So for instance, if we imagine some sort of a nucleus made of red and blue neurons that talk to another nucleus that is made of green neurons, and those mediate the response to a set of stimuli. You can imagine that if during brain development, the red and blue cells do not connect to the green cells, then you're not going to get a response. So, of course, in that sense, that the development of the brain is important for its function, but this is completely trivial. But there is a way in which this is somewhat less trivial, in that the connectivity between the red and blue neurons to the green neurons may develop in many different ways. And it is completely non-trivial and actually totally not obvious how the responses in each of these hypothetical cases, and of course hyper-simplified hypothetical cases, might differ from one another, and if so, why they would differ from one another. So therefore, understanding, in fact, the rules that allow the brain to develop the way it does would tell us something about what we expect for the brain to do, what constrains its capacity to respond, so what is allowed and what is constrained, and how and why are these constraints there, and how far do they go, and why isn't everything possible, and so on and so forth. So I would argue that just looking at the final structure, its activity, is not sufficient to gain deep enough insight into how the system works. Understanding how the system builds itself is part and parcel of understanding not just how it works, but why it works the way it works. And so if we want to understand something about how neural circuits might govern behavior and how variation in neural circuit architecture might govern variation in behavior, one needs to understand what are the rules that the brain utilizes in order to generate these neural circuits. And work on many systems, the vertebrate retina included, but a lot really in Drosophila and C. elegans, because they're very small, tractable, and so-called deterministic systems, has led to a paradigm that dominated the field for a long time. In fact, it all started with Roger Sperry's work on chemoattractivity, and finally his sort of formulation of an extreme version of chemoattractivity, which said that every neuron knew exactly what it was doing because it was equipped with the right molecular codes to find its partner. Studies on the sensory system in Drosophila, both in the eye and in the nose of the fly, which you see a schematic of here. So this would be, my mouse is not moving. Anyway, doesn't matter. What is called the antenna here is the nose of the fly, and the antenna lobe is the part of the brain where the nose neurons project. And there's been this idea that as soon as cells are born, as neurons are born, they're equipped with a specific fate, a specific identity, which here is reflected as blue, green, and red, for example. And the idea is that the molecular identity of the cell equips it with a translation of the genetic code that makes its wiring inevitable. That is to say that the green neurons will always wire in the green part of the brain, and the blue in the blue, and the red in the red. And it's a simple consequence of the fact that they're born that way. So the moment the cell is specified is the moment it has all the information necessary to find its target. Which of course means that there is no developmental plasticity, because there is a long time between the birth of the neuron and the time when it's finally synaptically connected to its target, and that synaptic connection is active. However, there is a paper that I think doesn't get often enough cited, which is work from a collaborative work from Li-Chun Luo's lab and Rachel Wilson's lab, that sort of challenges this idea. So what was done in this experiment that you see schematized here, and where you don't really need to worry about the details, is you just took one figure from the paper to illustrate the idea. What they did was they took a driver, an enhancer construct that labels about 15 interneurons in the brain, in the olfactory part of the brain. And what they did is they made 161 single-cell clones from this population of 15 neurons, which of course, if you calculate, means that they should see the same, on average, they should see the same neuron a little over 10 times, right? So you take 15 cells and you see them in 161 times in 161 different animals. So that, on average, should give you the same cell back a little over 10 times. Which means that in total, you should see 15 patterns of anatomy and 15 patterns of electrophysiology. What they saw, though, is not 15 patterns, is not 20 patterns, is not 50 patterns. They made 161 different clones, and they saw 161 different anatomies and 161 different physiologies, which means that the same cell was slightly different in each brain. Yet, of course, we know that the totality of the circuit anatomy is highly robust. I mean, there's no one who has difficulty recognizing what part of the brain is what, or where an axon from a retinal ganglion cell will end up in the superior colliculus, or etc. So how can there be variability at single neuron resolution? How can every cell be different in each organism, and yet the circuit overall highly stereotyped between organisms, right? So what kinds of rules allow variation at single cell level and yet force stereotypy at the circuit level? And of course, that means that if every one of us had a slightly different version of any set of given circuits, and yet kind of on average similar brains, then that begs the question of why and how are we, and therefore our brains, or I should say our brains and therefore we, different as individuals and yet stereotyped as a population, right? I mean, no one's going to ever confuse a butterfly with a honeybee, right? I mean, it's pretty clear. And so why is that? Obviously, species are species for a reason. They share a common genetic heritage. And so one can argue, and of course it is completely correct, that individual differences in the genetic code in your DNA sequence in our DNA sequences make us different, right? At the same time, we know that mosaicism, genetic mosaicism apart, we know that individuals who share almost fully or even fully, such as in parthenogenic reproducing species, their genetic code, their genetic sequence, are not absolutely identical. So we know genetic differences are super important, but we know that they're not the whole story. When it comes to brain and behavior, we also know that neural circuits are modified by experience. We don't know to what extent they're modified morphologically by experience. There's probably some level at which they are. They're mostly modified synaptically by experience, but that does modify behavior. And sometimes these experiences can be so strong and so important that they actually modify behavior for a very long time to come. I mean, anyone who's experienced a trauma or an extremely unpleasant sensation in a given context does everything possible to avoid that context for the rest of their lives. And that, of course, means that they change their behavior. So what those two things combine to make us individuals. But like I said, both of those things are controllable and we know from experiments on species where these things are controlled by definition because the individuals are genetically identical or because the organisms can be made to be genetically homogeneous, and where the environment can be controlled, even the microenvironment can be controlled during development, we know that these individuals do not develop in exactly the same way, and their brains do not either, and therefore their behaviors end up not being absolutely identical. Now, in the field of developmental biology, this is not a new idea. The fact that development is subject to what is called stochastic developmental noise, which results in intrinsic developmental variation, is kind of a common accepted thing. It's called that mostly because we don't understand how it happens, so we call it intrinsic variation and stochastic noise. And one can argue to what extent it's stochastic and to what extent it's noise, but it's definitely there, and it cannot be made to go away either by genetic homogeneity or by control of the microenvironment. The idea that this might be a contributor to the way the brain wires and therefore the way behavioral variation might emerge is somewhat, or was at least a little bit less accepted of an idea. And so and that's basically what the story I'm going to tell you about today is. And so just by way of introduction, we're going to be using the fly brain, which you see to the right here, parcellated into its different anatomical areas, just like our brains are parcellated into different anatomical areas that are that do different things, or at least privilege doing different things as opposed to others. We know that, in V1 mostly you do vision, you don't do olfaction, for example, and so on. And the same is true in the fly brain. And this part that you see squared here or rectangled in white here is the part of the fly brain that does vision. And the fly, and these are called the optic lobes, and the fly dedicates about half of its brain, if not more, for visual processing. So within the optic lobes, we're going to be studying a set of neurons, called the dorsal cluster neurons. And we're going to study them as a wiring model to try to understand how neural circuits develop and how the rules that govern neural circuit development might allow population-level stereotypy and yet individual variability, or single-cell variability between individuals. Now, the dorsal cluster neurons are a really cool model to address this question because they're sort of intrinsically highly variable. So there are two clusters of cells on each side of the brain, as you can see here. And the number of neurons can vary between 22 and 68. These neurons will cross the brain from the left to the right and the right to the left. So they're contralateral, higher order visual interneurons. And when they cross, they will make it all the way to the optic lobes. And in the optic lobes, they will make a choice between a distal target called the medulla, or abbreviated as ME, and a proximal target called the lobule. Sorry, I went too fast there. Oops. Okay. We'll get back to that in a second. Now, in the medulla, this distal target, out of the 22 to 68 neurons, 6 to 23, depending on what brain you're looking at, will make this choice, okay? And it turns out that this number, there's no correlation between the number of axons that end up in the medulla and the number of neurons from which they originate, which tells us that this is not a simple sort of, the fewer, there's some ratio, constant ratio of cells that is going to go to the medulla, and it's a fixed sort of a Planck's constant type ratio, and you just multiply by the number of cells with this constant, you get the number of axons in the medulla. That's not the case. So how does it work then? How does an axon from one of those neurons make a choice between going distally or staying proximally, i.e. how does it choose between the medulla and the lobule? One idea is maybe not all these axons actually cross the midline and go to the other side. Maybe some are ipsilateral and others are contralateral. And we ruled that out by showing that actually every single one of these DCN cells, I don't know why that went back, sorry about that. Every one of these single DCN cells crosses the midline, and it does so because it's able to ignore negative signals from neighboring neurons. And one of those negative signals is a protein called SLIT, and its receptor is called ROBO. I think those of you who may have attended last week's talk may have heard about ROBO. And so the axons, the DCN axons do not express ROBO, and so they can just cross the brain happily, get to the other side. They express a receptor for a protein called netrin, which is an attractive cue, and which is expressed in the optic lobes pretty broadly, and so these axons innervate the optic lobes. And we can show this because if we express ROBO in the DCNs, we make them all go contralaterally, as you can see on the bottom left. Ipsilaterally. So we can make them turn around at the level of SLIT expression and go back to the same side. So initially, under normal conditions, all the cells go to the other side. You can make them all switch to one side. And therefore, the difference between the individual cells choosing to go to the medulla or the lobula has nothing to do with whether they cross to the other side or not. Okay, so that's not the solution. So if they all cross to the other side, they must, at some point, make a choice between the proximal target, shown in here in red, the lobula, and the distal target, shown in blue, the medulla. Okay, how is that choice made? And how can explaining that choice tell us something about the variation we observe in the wiring of this circuit. So how and when do these two DCN subpopulations emerge during development? Now, this is published, so I'm just going to give you the upshot of the story, published a long time ago now. It turns out that the DCNs talk to each other via the Notch pathway, via a process called lateral inhibition. And the interesting thing about lateral inhibition is that it is an intrinsically stochastic process. It is genetically encoded to be stochastic, all right? So, and this is, as an interesting aside, we tend to often sort of mix when talking between genetic encoding and genetic determinism. But that's not the same thing. Something can be genetically encoded and yet not deterministic. As, for example, the way the Notch pathway works. It's completely genetically encoded. It's absolutely not environmental, and yet it is stochastic by definition. And so the way it works is cells talk to each other via this Notch signaling pathway. And if you don't know this pathway, don't worry about it. It doesn't matter. And basically, if they have Notch off, they go to the distal target. And if they have Notch on, they go to the proximal target. And it turns out it doesn't matter which cell, and I'll show you an example of this in a moment, which cell is Notch on, Notch off, as long as 1 in every 5 or 6 cells on average is Notch off and the others are Notch on, you get this, this effect. If you inactivate Notch, you send more of the axons out towards the medulla. You can see this in Notch loss of function panel. If you activate Notch in all of the neurons, then none of them go to the medulla. So the levels of Notch activity in each cell are necessary and sufficient to determine whether it's going to be a distal target axon or a proximal target axon. And because the process is stochastic, you get this sort of variation from brain to brain. But how exactly and when exactly does this happen? When is the decision made, and how does the decision actually translate into wiring? And oh, sorry, before I get into that, let me just share with you what I mean by it's a stochastic process. Okay, this is frozen. There we go. And how we sorted it out in a schematic. Again, this is all published, so I'm not going to go through the data in great detail. But basically, the basic idea here is under wild-type conditions, a few cells, shown in green here, will talk to one another, and one of them will be the winner. And that winner will be the Notch low cell, and it will go to the distal target. All the cells that are in direct touch with it will stay in the proximal target because they've lost, until the next cell that has no interactions with that one red cell has to make a decision. And in this subset of cells, again, you get another axon and another axon, another axon. And so this explains why the pattern is sparse and why there are specific distances between axons in the medulla. You can demonstrate that that's what's going on by making individual cells mutant for Notch. And in that case, it is that cell that you made mutant for Notch that will win the competition and send its axon to the distal target. And the other cells will retract in response. So you can sort of tweak the system where you can choose which cell wins. That automatically makes all the other cells, quote unquote, losers. And so the rare cell that you made Notch mutant will go to the medulla, all the others will be in the lobula. But the pattern, the overall pattern, will actually be normal, will be within normal range. And that is the demonstration that it doesn't matter which cell goes to the medulla because it simply is compensated for by all the other cells. And you can show that's the case by basically making a bunch of neurons mutant for Notch so that they cannot interact with each other. They all think they're winners, and in that case, you send them all to the medulla. Okay? And so that's the demonstration that it's mutual inhibition at a local level that is making a cell-by-cell decision on a stochastic basis. So then the specificity of the wiring pattern, when we look at 100 flies, dissect their brains and say, well, there's on average that many axons and this is the spacing between them and so on, we look at something that looks highly stereotypical with some error bar in between that we tend to ignore for some reason. Well, it turns out that the error bar is what is telling us about how the process works. That is to say that what we would refer to as wiring specificity is not a priori encoded in the cells, in each cell individually. It's an emergent property of the cells interacting with one another, making stochastic decisions, and interacting as autonomous agents. Okay? So how do we get there? How do you go from, so this is the theory, right? You have Notch, and Notch inhibits somehow the growth of the axon. And so you have to have Notch low in order to go to the medulla. Okay, great. How does it really work? And Maeva Andrietsilavo in the lab, a very talented postdoc in the lab, has been tackling this issue. So what I'm going to show you now is some unpublished work from Maeva, where she used the technique developed in Robin Hiesinger's lab of intravital imaging. So what we basically do, what Maeva does, is she's able to image the brain of the developing fly during development, watch these axons grow out, and then keep that animal alive until it becomes an adult, and go back and match back every single axon that ended up making the distal target selection, where it was during development, and at what moment it made the decision to grow or not to grow, and what it looked like at that point. So that tells us what do those axons that are Notch low look like compared to the axons that are Notch high. She can do this at the population level, but she can also do it at single neuron level by making clones, which you see here. So what you see in the upper panel is, over time, the imaging of a single axon that actually is going to end up in the medulla. And in the lower panel, the imaging of a single axon that is not going to end up in the medulla. And what Maeva found is that the axons that will protrude past the initial target to the distal target develop this bizarre looking bundle of fibers. It's as if it's growing 3 or 4 parallel axons together. So you have a single initial shaft, and a number of the short filopodia decide that they're going to make sort of new axon shafts and develop these parallel fibers that are going up. No, they're not climbing, they're going forward towards the distal target. And once the distal target is reached, all of them will disappear and only one will remain of those. So it's like the cell itself creates competition within its own axon between multiple fibers. Whereas the axon that will not go to the distal target, you can see, remains locally exploring and does not develop these long bundles of parallel fibers that are. So now we have a pathway and we have a phenomenon. And so we have a decision maker and we have a phenomenon that is associated with that decision. Are they connected to one another? And the answer, again, I'm sorry I'm giving you this as schematic in the interest of time, but basically, the basic idea is it turns out that there's a critical period in which Notch activity is necessary and sufficient to suppress bundle formation. So the neuron that has low Notch will not have bundle formation suppressed and will make this bundle. Now, this bundle is an actin-rich bundle of very long parallel axons that come from the same axon, parallel fibers that come from the same axon. And they last just long enough to allow one microtubule to invade one of them. So it's like bet hedging. You bifurcate or trifurcate or quadruple furcate your axon. You make all of those actin-rich fibers grow, and one of them will be invaded by a microtubule. It doesn't matter which one, because it is that one that turns out to be the final axon that will innervate the medulla. So now we have a translation from a stochastic decision at the sort of conceptual level, at the command and control level of the cell, of the nucleus, to the execution of this process also in a bet hedging scenario, where you just allow things to grow and hope for the best. And of course, microtubules grow, that's what they do. And so they'll grow into one of those things and turn it into a distal targeting axon. If you activate Notch early enough, you can suppress all of this. If you activate Notch after the bundling has happened, there is no effect. So there's a critical period during which this decision has to be made, and it gets translated in the way that I've just described. So that solves the problem of how the pattern arises during development. And that again tells us, therefore, that wiring specificity, at least in higher order circuits, is a problem of pattern formation. It does not require pre-configured address recognition codes. Those codes can actually emerge as cells interact with one another, interact with their environment, but also interact with themselves in a way, as I just showed you with this bifurcation. That the noise intrinsic to molecular processes, because all molecular processes are noisy and variable between cells, but this is not a problem that biology is trying to eliminate. This is actually a feature of the system. It's not a bug, it's a feature by which the algorithm that regulates brain development works to generate variation in the neural circuit, but because the different cells talk to each other and inhibit each other from doing the same thing, you get this sort of stereotypy at the population level. And the sort of conceptual correlate of this, when we think about the fact that the brain is not designed by an engineer, it wasn't created by some supreme deity or whatever, it is an evolved entity from a pre-existing evolved entity. One must, of course, put everything in the context of evolution, because nothing makes sense in biology except in the light of evolution. One begins to think about how these processes of variability and noise might actually contribute to the robustness of the development of the brain and allow further evolvability of a very robust structure. And we can discuss that at length if you like afterwards, but you can also read about it in opinion papers that Robin Hiesinger and myself wrote, but many other colleagues as well, Ben de Beauvoir and many others, Mark Kirchner and many others have written about these ideas. Okay, so what do we have? We have a circuit that's different between different individuals, and we have the potential, therefore, that the way this circuit is wired, if you think back at the initial diagram I showed you to argue that the importance of brain development to brain function is not just trivial, we can really begin to ask about how these subtle, quote-unquote, variations in circuit wiring might actually contribute to the emergence of personality traits, by which, of course, very operational, down-to-earth definition, the simple idea is a personality trait is something that defines you as an individual because it's invariant in you and dissimilar from the others, okay? So you are similar to yourself over time, but you're different from others, okay? So do Fly show this? And if so, can we use the DCNs as a model to query this question? Now, to begin thinking about how that might be done, it turns out that there are clues in this in the classic literature of Drosophila visual neuroscience. One of the founding fathers of the field, Carl Georg Goetz, who worked on visual behavior in many insects, and his students then wrote the following, or I'm paraphrasing, but basically wrote the following insight. And I have to say that in going back and reading those papers, at least those of them that were written in English, because some of them were written in German, I couldn't really figure out exactly where that insight came from, exactly what behavioral elements in the behavior of an animal led them to this sort of strongly formulated conclusion. But nonetheless, there it was, in which they suggested that when a fly is moving towards an object, so one of the things that a visual system does is react to motion in the environment or fixate on an object, right? So I think it's obvious. All visual systems have this innate capacity to either track motion or to do object fixation, orient yourself towards an object of interest. And what Goetz and his colleagues hypothesized is that the ability of the fly to orient itself towards a fixed object depended somehow on a qualitative asymmetry in perception between the left and the right eyes, or the left and right sides of the visual system. They suggested that if there's asymmetry in this perception, as the fly turns or walks towards an object, if the object appears differently on one side than it on the other, then somehow that allows the animal to put it better in the front of its visual field and to orient better towards it. Okay? Again, no idea why they said that, but that's what they said. And what's striking is that about a decade later, Martin Heisenberg, another giant in the genetics of behavior, particularly in the Drosophila visual field, was studying object orientation as independent from motor perception, from optomotor behavior. And he and Reinhard Wolf hypothesized the existence of a higher order circuit that was a contralateral circuit that mediated crosstalk between the two visual systems that was required for object orientation. This was a completely hypothetical construct based on the fact that if they blinded motion vision, the flies could still orient towards an object, which suggested to them that object orientation did not require each eye separately, but a communication between the two eyes. Okay? And for that communication, they said, they drew, in fact, a hypothetical circuit that communicated the information from the left eye to the right eye. Now, let's take a second here and think about this together. The basic idea is, if your perception of an object you're moving towards is asymmetric on the left than it is on the right, then your global perception of it is more accurate. Your global perception of its position relative to your motion is more accurate. That's one idea. The other idea is that object orientation requires a contralateral circuit that communicates between the two sides of the visual system. And of course, you know, it's fairly obvious that what the DCNs are a higher order contralateral circuit that communicates between the two visual systems of the fly, and that is variable not only between every individual because of the way they wire, but also between the left and right sides of the same individual, which, of course, variation between left and right is, of course, asymmetry. And so we started to look at whether, indeed, not only is the DCN circuit variable between brains, but within the same brain. If these are really autonomous decisions made by each cell on the left versus the right independently, then we should have differences on the left and the right, and that is exactly what we see. And if we now do transsynaptic tracing to ask whether the asymmetry in the wiring of the DCN axons results in the asymmetry of the wiring of the circuit, then the obvious answer, I guess predictable answer, but nonetheless nice to see answer, is that that is the case. So when the DCNs, for example, do not wire up to the dorsal medulla on the right, the dorsal medulla cells do not receive compensatory input by some branch coming from the ventral axons or whatever. They're really not connected. And that, of course, gives rise to asymmetry. So that now allows us to test, is it true that asymmetry is required for object orientation? And does the variation in this asymmetry produce variation in object orientation? And so now we need an assay for object orientation. And again, we go back to one of the classic experiments done by Götz and his colleagues, which is called the Buridan paradigm, or the dual stripe fixation assay, in which you put a fly in the middle of an arena. That's what you see on the top left, schematized. The bottom left is the real picture of one of those arena in the lab. And you put that fly in a completely lit drum barrel, in which the fly has at 180 degrees 2 dark stripes. And the fly cannot reach the dark stripes because the platform on which the fly is walking is surrounded by water, and the flies can't swim. And they can't fly because we cut off their wings. So now what the fly does, it spends its time walking back and forth, back and forth, back and forth, trying to reach each of those stripes. And I'll show you an example of that in a video. You will see Garrett, there, the postdoc who did all this work, dropping the fly on the arena. The fly tries to go to the first. stripe, can't, goes to the other side, explores the possibility of crossing the ocean to get to the dark stripe. It can't, so it comes back, then changes its mind, goes back, tries again, and then says, okay, there's no way, I'm going to go back to the other stripe, and basically back and forth and back and forth and back and forth. Yeah, they are that dumb. So, so basically what you can do is you can, of course, test the same fly over and over and over and over and over again, over days and weeks, and you can transform the path tracking of the fly into a heat map of its location. And if the fly were to walk randomly and differently on each day, then the heat map would fill the entire arena. But in fact, what the fly does on day 1 is what the fly does on day 2 and on day 3, and that is reflected in the heat map of its location over many days and weeks. And so here, for example, are the heat maps of 3 individual flies over a period of 3 days. So we test them, we rest them a day, we test them on the second day, we rest them again, and we test them again. And so this is what individual 1 looks like, individual 2, individual 3, which you can see has a complete preference for walking between the stripes on the left side. Individual 4, who really avoids the middle and walks through mostly on the edges, and so on and so forth. And now you can ask, what is the correlation between the behavior on day 1 versus behavior on day 3? And you can do that again 2 weeks or 3 weeks later. And you will find that basically individual flies are highly faithful to their own behavior. So this is an innate trait, essentially a personality trait of the fly. Its object orientation is a personality trait that is different between flies, but the same over time in the same fly. Is this heritable? The answer is no. So you can take 2 flies that like to walk down the middle and cross them to each other, and they will have all kinds of babies, which you can see the distribution there in the histogram. And they will have babies that walk like them, right through the middle, but they will also have babies like individual 3 who really look like the neighbor's kids. And so that walks all over the place. You can do the same experiment, taking flies that like to walk along the edges, mating them. Again, you will get a distribution with a mean, so the population mean is always the same, and yet the individual variability doesn't differ. You can do that with strains of flies that have been inbred for 20 generations, and it's the same story. You can do selection experiments in which for 10 consecutive generations, you will take the flies that like to walk through the center and breed them again and again and again and again. In every generation, you regenerate the entire variance of the population, just like you see here. So it is not possible to breed out the variability, and it is not possible to make sure that all your offspring inherit exactly your behavior, no matter how many generations you try to do that for. So now we have a circuit whose variation is a product of genetically encoded stochastic processes that basically shows a range of asymmetries between left and right across individuals. It's a visual circuit. It's contralateral visual circuit. And we have an object orientation behavior that shows variability between individuals, that shows basically personality traits. So is there a correlation between the variation in anatomy and the variation in behavior? And the first question, and so to address that, the experiment, the heroic experiment that Garrett and his colleagues did, was to record the behavior of about 300 flies, then dissect their brains, record the anatomy of the DCNs in each of these brains, many aspects of the anatomy, as you can see here, and try to find correlations between individual behavioral parameters and individual anatomical parameters. And from this unbiased approach, what emerged is that when the DCN wiring is asymmetric, the flies walk a straight line between the 2 objects down the middle, and when the DCN wiring is symmetric, the flies walk all over the place. In diagonal lines, they meander, and so they have what we would call a high object deviation index, whereas the asymmetric flies have a low object deviation index. Okay, but do we even know that the DCNs are required for this object orientation behavior? And the answer is yes. So here's the correlation between anatomy and behavior on the correlation plot on the lower left. Well, my lower left. If you silence the activity in the DCNs, so you let them develop just fine and you silence their synaptic release, what happens is that all correlation between behavior and anatomy completely disappear. Okay, so the DCNs are required for this behavior, and their asymmetry correlates with the. You can predict how an animal will behave if you could see the DCN wiring. You could also predict how asymmetric the DCN wiring is from the behavior of the individual. Now, this is nice. The DCNs are required for the behavior. Their asymmetry, which is the product of their sort of stochastic developmental processes, correlates with the individuality of the behavior. Is it actually causal, right? And so what we did in these experiments is, in this particular experiment, is an experiment where we basically block the ability of the DCNs to talk to each other. And when we do that, we block notch signaling. And when we do that, we send the majority of the axons to the medulla on both sides. So we start to create a population in which the DCNs are much more symmetric on the left and the right. And when you do that, you totally create a shallower correlation plot, but you preserve the correlation. So the population as a whole shifts to more symmetric behavior, as you would predict, meaning that they meander all over the place. But the correlation between each individual and its behavior remains exactly the same as it is under control conditions. Okay? So basically, we can rewire the circuit to make it, on average, more symmetric. And we can get the population to behave, on average, more symmetric. And yet every individual, when you dissect the brain, is still faithful to their wiring path. The behavior of each individual is still faithful to its wiring path. Now, recall that the initial insight of Guts was the idea that perceptual asymmetry would create better object orientation. And so what we wanted to test is, what we're suggesting here is the asymmetry of the DCNs that essentially regulates the quality of object orientation. The more asymmetric the DCNs are, the more asymmetric the information flow is between the two visual systems, and therefore the perception of the organism of this, this asymmetry between the left and the right is a perceptual asymmetry that's regulated in the brain, not in the eyes. The eyes see the same thing. It's the brain that sends the information in an asymmetric way, allowing the animal to focus better. But if this were true, if the fundamental insight were true, the source of the asymmetry in perception shouldn't matter, right? And so to test this idea, we took individuals that are highly symmetric in their DCN pattern, and we can tell that because the individuals have bad object orientation. They meander all over the place. And we blinded one eye in those individuals, right? With the idea that if you take individuals whose eyes and brains are symmetric and you blind most of one eye, then you create visual asymmetry at the sensory level, not in the brain, but in the outside. And what happens when you do that, as you can see here, is that the same individual who initially was meandering all over the place between individual 1 in orange, meandering all over the place between the two stripes, now walks a much narrower path between the two stripes. They still have a highly angular motion, probably related to the fact that one eye is blind, but they actually walk a narrower path. The same is true for individual 2, and you can see on the plot below the population plot in general, if you take the animals that have very high stripe deviation, object deviation indices, and you blind one eye, the overall population drops and becomes a better object-orienting population. So that is it. That suggests that not only were both Guts and Heisenberg right, but in fact, it so happens that for the last 20 years or so, my lab has been working on the neurons that were predicted to do this at least 30, 40 years ago. Okay, so I'll stop here and just conclude with a few take-home messages. The first is that as far as higher order circuits in the brain, at least as the DCNs as a model are concerned, we don't need to worry about how much every cell knows and how the genetic code works and in sort of a cascade of transcriptional events to tell every neuron exactly what it's going to do and which other neuron exactly it's going to wire to. I don't think that model is plausible. I think it's quite wrong, actually. But at least as far as these neurons are concerned, the specificity is an emergent property of a pattern formation process that simply is filled with cell-to-cell interaction events within critical periods, such that something has to happen at a given time point, because once it happens, it creates a new state whereby the neuron cannot go back to the previous state. That the noise that is intrinsic to every molecular process, from transcription to splicing to RNA stability to translation to protein folding, all of that, these are not bugs of the biological system. This is exactly what allows biology to not be physics and not be chemistry. That is to say that it's a messy, natural selection-driven process. So noise and stochasticity are features of the biological system, and they are features because these are not design problems. These are evolution problems that have to deal with unpredictable events in which certain individuals will propagate the species and others will not. And that this actually makes the development of the brain far more robust. And there may be a price to pay in individual variation, in that you have greater susceptibility to certain problems, the likelihood that any given individual may or may not end up being a prey or whatever, right? But it does make the process as a whole, the process of brain development and the control of behavior, a much more robust process, and therefore a much more evolvable process, which is, at the end of the day, the only thing that natural selection really cares about. So obviously, I get to present this to all of you. I didn't do any of this work. The work was done by an incredibly talented postdoc, Gerrit Linneweber, joined later by another super talented postdoc, Mayeva Andriatsilavo, that I showed you, is doing all the live imaging on when the decision is really made, and a PhD student, Suchetana Biasduta, who is working on how the DCNs actually establish synaptic connectivity and what the link between axonal branching and synaptic active zone formation is, and whether there's a feedback or a separate control or whatever. And hope to tell you about that in some other occasion, maybe in 3D in a few months. So that's it. Thank you for listening. Thanks to all the people who've trusted us over the years with funding. And I'm more than happy to take your questions.
All right, Bassem, thank you very, very much. According to the now tradition, I'll be clapping physically, but if you go on your crowdcast screen, you'll see comments from people and applauses. So yes, thanks a lot for driving us through this and this particular last set of experiments, which I find are truly fascinating. So we have a couple of questions. Feel free to continue voting. Also, the students that are out there take also the opportunity to ask questions in a relatively anonymous way. It's a good exercise. So let's get started, Bassem. I have the questions in front of me. First question by Isabelle Caillé. What is the origin of the differences in notch levels between neurons?
Right. So that's in a way the beauty of notch signaling. Can you hear me, Denis?
Yes, it works. Yeah.
Yeah. I mean, in a way, that's the beauty of notch signaling. You don't actually require initial differences in the sense of differences in levels of expression regulated by different enhancers or whatever between cells. Because of the way lateral inhibition works, whereby essentially initially equivalent cells are express both delta and notch, so the ligand and the receptor. Any small variation that is due to stochastic noise, differences in intracellular degradation of delta or notch, how much receptor versus ligand happens to be at that moment on the surface of the cell, these small differences get amplified because it turns out that as soon as you activate notch in cell A, notch represses delta expression. So as long as the levels are roughly equivalent, there is this sort of dynamic equilibrium. But as soon as one cell signals a little more to the other cell, the increased activation of notch in that cell suppresses delta, which allows it to signal less back, which means now you go from equivalent signaling to a slowly amplifying difference in signaling. And as soon as that is established, it's irreversible. Okay. And that's how we think it works.
Okay, thank you. Next question: Do you think inter-individual variability in behavior is truly higher in animals with bigger brains, and if so, which types of mechanisms would underlie this?
I don't think so. I don't think it would be higher in bigger brains. I have never seen evidence to convince me that, for instance, if you take C. elegans with 302 neurons, that we are told are completely invariable between individuals. So if there is some level of variation, it's very, very small. When you look, on the other hand, on the spread of the data points in any given behavior, you compare a mouse experiment to a fly experiment to a C. elegans experiment, you don't really see differences. So I don't think that it's the size of the brain. I think there's a constraint on how much outside a certain distribution you can go and still be viable as an organism. How far can your brain deviate from the mean of the species, of the population, and still give rise to an organism that is viable? And I think that is where the constraint is. And I think that constraint is probably very, very similar across animals. Now, what you could imagine, on the other hand, is that different behaviors could be differently variable. That is to say that in response to certain things, where you really have a small number of cells in a labeled line sort of approach, you would really have most of the population around the mean. You would have a narrow distribution. Whereas where things, if it's a behavior that was not correlated directly to the way the species exploits its niche essential for survival, you might have greater flexibility in behavior. So in those types of, I think it's harder to say whether sort of what we might call higher order derived cognitive tasks that were not essential for the emergence or survival of the species, but then became sort of spandrels of the fact, like in us, the fact that we have this sort of big brain, allows us to do things that were essential, not essential for us to emerge as a species or survive long enough to stay. That within those sort of derived abilities, maybe in cultural behaviors or whatever, maybe there's greater variation.
But within a given individual, would you say that there's less variability if the circuit is smaller, whatever that means, if there's less degrees of freedom?
I'm not convinced about that, to be honest. And the problem, of course, the problem with that is, every behavior, even if the initial circuit is driven by a single cell, right? A single sensory neuron that is receiving the information, the moment it gets into the CNS, that information starts to get distributive. And we know from electrophysiological recordings in any system, and many of you know a lot more about this than me. I mean, if you stimulate a given neuron, right, there is also a sort of a ripple effect in activity that is a function of the distance of connected neurons from that one neuron. That is to say that the closer you are, the higher you're likely to be connected, and so the higher your probability of response, and the further you are, the lower your probability of response. So the moment you send information to the brain, you're creating ripples, which then create other ripples and other ripples. Mm-hmm. So even if the sensory pathway and at the other end, the motor pathway, is extremely constrained and labeled, everything that happens in between, this hidden part that I showed in slide 1, makes me think that it's almost like the brain is evolved to be a random number generator and a variation generating machine, and that gets constrained by sensory and motor, and not the other way around. It's not the brain that constrains our behavior; it's the periphery that constrains our behavior. In my opinion, I mean, this is wild speculation.
Interesting. Thank you. So next question, how does evolutionary selection pressure affect inter-individual viability? In other words, when condition gets harsh, quote unquote, would that tend to decrease or increase viability, genetic, behavioral, or otherwise?
Right. I mean, that's a great question. And of course, it's not my expertise. I think a couple of weeks ago, there was a speaker from the Karolinska who's really the expert on that. But I can speculate a little bit, and we've done recently. So maybe I'll rephrase the question. So I don't know is the simple answer, right? The less simple answer is if a given genome, as I argued and I showed you data, in fact, if a given genome can generate multiple examples of a brain, right? So variation, I mean, the individual variability of the brain means that basically it's multiple examples of the same brain, right? So variants of the same brain. That means that if one genotype can generate multiple phenotypes, it also means that the same phenotype for any given circuit can come from different genotypes. Okay? Mm-hmm. So even though the type of variation I'm showing is non-heritable, and therefore you would say, well, if it's not heritable, how can it be selectable? It's true. Any given phenotype is not selectable. But a genotype that encodes a specific phenotype that selection acts upon is selectable. And since multiple genotypes can encode the same phenotype, even if selection is extremely high, selection pressure is extremely high, there will still be multiple genotypes that pass that bottleneck because they all contribute the same phenotype, right? Mm-hmm. And so in a way, even though you would predict if you had a one-to-one genotype to phenotype map, that high selection pressure would result in extremely low genetic diversity at the end of the bottleneck. In fact, what we're suggesting is it doesn't have to be that way because multiple genotypes can survive the same selection. And that would explain why, there's no way you could get around genetic diversity, and there's no way you can get around phenotypic diversity, no matter how you shift the selection pressure. Mm-hmm. On a quantitative level, of course, depending on how strong the pressure is, you're going to have some trade-offs. But that's really very far from my area of expertise, and I'd rather not speculate beyond what I've done so far.
Okay, thank you. How does nuclear notch transcription and signaling influence the distal axons in a very time-sensitive manner?
Can you repeat that, please?
Yeah. How does nuclear notch transcription and signaling influence the distal axons in such a time-sensitive manner? Yes,
that's great. So the fact that notch activity has a critical period actually may not require a magical solution. In fact, in response to the first question, I described how lateral inhibition works. And so basically, as soon as you create this differential, that's it. The different cells are different and are no longer responsive. It is not possible anymore to change them under wild type conditions. And so it turns out that, and we're still looking at that, we don't have all the answer, but it looks like notch activity in the nucleus changes the regulation of genes that are involved both in actin regulation and in microtubule regulation. So there seems to be a feed forward onto RAC activity, RAC GTPase, and onto JNK signaling, both of which, one is well known, well, both are major regulators of the cytoskeleton. So we think that as soon as the Notch state in a given cell is established, the consequence is that there's going to be greater actin depolymerization and repolymerization, allowing those filopodia to go crazy and allowing, therefore, microtubule extension. And we think that JNK activity might also stabilize growing microtubules through phosphorylation, indirect probably, to allow these axons to grow. So that's how we think it's translated. What is interesting is that even if we try to change the state of Notch activity after the critical period, so if we impose high Notch on all the cells after they've made the bundle, they no longer respond. And that's interesting, and we don't know why that is. It's possible that that might be because the downstream events have already kicked in, and you can't switch them back. It might also be that, of course, some epigenetic state has been imposed by Notch, where the nucleus, the gene expression program, is no longer responsive, and there's nothing to do. And those are interesting questions to explore further.
All right. Next question. Is there evolutionary pressure on the Buridan paradigm behavior? Is there any advantage for the fly to walk straight or not? Do you think that this is the same for flying flies?
Sure. I mean, it's a completely artificial system. And just because the DCNs seem to regulate object orientation in this particular artificial assay, I cannot guarantee you that that's what they are doing in the natural fly in nature. I have no idea. It tells us something about what the brain could do. Of course, it doesn't tell us about what the brain really does, right? Yeah. In real life. Now, does object orientation in and of itself matter? And the answer to that is obviously yes. I mean, a trivial answer to a trivial reason why it's yes is because it has survived selection in all visual species. So it must be important for something. And the other thing is, of course, if you cannot properly orient and alter your motion towards an object in response to what else is happening in the world and in response to your own motion, how can an animal ever reach a source of food, a potential mate? How can a goalkeeper, track the movement of the ball and leap just in time to grab it? And we all know that without football, life doesn't mean anything. So yeah, there is evolutionary pressure.
Okay. We have a question by Shuba Tore. Hi, Shuba. She's wondering whether the flies could actually be trained to walk straight, basically.
Fantastic.
That would mean this particular circuit can be overridden by other circuits.
Yes. So I think there is good reason to say yes, because we know that innate preferences that have to do with other types of behaviors can be overridden by learning, to at least to one extent or the other. We haven't done the experiment yet. We're gearing up to do exactly that, which is to see if we can use associative learning conditions, so associative conditioning, to trick the flies to want to walk more straight, and see what that implies to what other circuits might be implied in this and what the plasticity mechanisms might be. But at the moment, it's just an idea and a project we have, and we don't have any answers to that.
Can you train them to stay at home?
Well, yeah, unfortunately, we confine them by definition, right?
All right. Maybe related to that, does neuronal activity play any role in the wiring of this circuit? There's a few questions on the role of activity.
Yeah, we haven't seen any evidence that activity is required for any aspect of the morphological aspects of wiring that I described. Even the number of presynaptic axon branches seem to be normal in the absence of synaptic release. So if we use tetanus toxin, for instance, a genetically encoded tetanus toxin light chain, we still get, a normal pattern of development. But we don't know whether later on, if you silence activity, whether there's going to be some sort of a degradation. I assume there could be a degradation. But the developmental aspects, at least at the resolution that we have looked so far, do not seem to be to require activity.
Okay. What happens when two or more distal axons target the same cell during branching? What determines the winner in this case?
Two distal axons target the same cell.
Yeah, I guess it relates to, I imagine the question refers to the resolution in terms of axonal specificity and competition. Like, would they compete for a region or a single neuron or maybe even a subcellular compartment?
We don't know for sure yet. So what Sushetana, Mayeva and Garrett in the lab are doing, and Sushetana mainly leading this effort, is to map at light microscope level using this technique called transtangle, which I briefly showed you, these red-green asymmetry, right? So this technique allows us to label all the putative postsynaptic cells, and then we can do that very sparsely across tens or hundreds of individuals to get a survey of all the postsynaptic DCN cells. So we're doing that at the moment. Each DCN axon has pretty long sideway presynaptic branches, and so we don't think that every axon connects only to a single cell. And in fact, it looks like every cell type, or a significant number of known cell types in the medulla, get contacted by DCNs, suggesting that they are not really that discriminatory in the relay. It looks like they're relaying global information from the left side to the right side to a plethora of medulla subtype neurons, which would then relay them back into motor control centers, optomotor centers, and eventually motor control centers. We don't think there's that much specificity at that level, but we're not sure. Garrett is doing EM level map tracing with the colleagues at Genelia, and we will see in a couple of years what the answer is.
Okay, perhaps also related to that, how do you come down on the common wisdom, quote unquote wisdom, that invertebrate neural circuitry is much more deterministic than invertebrates? Like, it's a little bit different than the question before. It's not about whether, small brains imply stereotypic behavior, but whether the circuitry itself is more determined.
I think, honestly, I mean, maybe I will be controversial here, but I don't care. Honestly,
it's recorded forever on YouTube.
Yeah, that's fine. I mean, my opinion is written and is recorded in writing already, so it doesn't. No, I honestly don't understand the insistence on this model. I don't understand the logic that, because they're smaller, they're more deterministic. Because the idea that something is deterministic or not, right? In principle, I mean, if it's deterministic, then it's in the code that creates it. It's not in the thing itself, right? What's deterministic is the process that generates the phenomenon. So why should it have to do with how big the animal is going to end up being? This doesn't make sense. I mean, either the genetic code and the way the genetic code is transcribed and translated are deterministic processes, or they're not. I would suggest that this question, right, if we imagine, at the beginning of life, there was a choice between making life deterministic and making life probabilistic, that would have been settled way back then. And from that moment on, I mean, how is it, if we all have a common origin, how is it possible that, some branches of life went with the deterministic path and the other branches of life went with the undeterministic path, and that has something to do with how big the animals ended up being? Does this mean an elephant heart develops less deterministically than the heart of a mouse. I mean, so there is something irrational about the idea in my view. So
well, couldn't it relate to the number, for example, of cell divisions involved? Like if you have a probability of something happening, the more time that particular event happens, then it happens, right? So different. Bigger brains is more divisions.
But that's different. That means that the outcome of the process may be more or less constrained. It doesn't mean that the process. So if we say the number of cell divisions, is noisy and probabilistic, then the number of cell divisions is noisy and probabilistic. Whether that number is 1 or 100 doesn't change the fundamentals of the process. What is different is if the progenitor, let's take a fly neuroblast and a human radial glial cell, okay? They will divide a different number of times. So of course the outcome of this is more likely to be variable in a human, the number of progeny that a human radial glia will generate, the spread, is likely to be higher in number than the spread that a neuroblast will generate. That doesn't mean that the fundamentals of the process of regulation of cell division is deterministic in the fly and non-deterministic in human. Yeah. That's absurd.
I agree.
That doesn't make sense to me.
Yeah.
I understand why there was this drive to suggest that brain wiring is deterministic, because when people were looking at relatively low resolution, they could see that the same axons wired to the same places, etc., all the time. And so if the outcome is quote unquote invariable, then the process underlying the outcome must be deterministic. My answer to that is, one, when we look at higher resolution, we see that the outcome is not invariable. And two, so there's a data answer to that question, suggesting that the idea is in and of itself not correct. But there's also a logic to it, which is very simple. It's non-deterministic processes can generate both variable and invariable outcomes. If a cell divides stochastically, but only once, it will always make two cells, 100% of the time. Okay, the outcome is invariable, the process is probabilistic. So, whereas deterministic processes, except for a couple of mathematically theoretical possible things, like class 4 automata, but anyway, deterministic processes, in principle, can only generate invariable outcomes. So if the outcomes are variable, how can the processes be deterministic?
Mm-hmm.
To me, it doesn't make sense.
Mm-hmm. Mm-hmm. Okay. We have a question from Alain Chedotal. Do you think such a process might contribute to handedness in humans? By the way, Alain, I hope you appreciate the haircut.
Your haircut, not mine.
I don't know. He didn't comment on yours so far.
Yeah.
So handedness. Do you think there's stochasticity and handedness in humans?
I mean, why not? It's an asymmetry, right? It's possible. I have no idea. There's also something, I mean, that's one possibility. There's something very, very interesting that we're working on with a colleague here in Paris called Mark Wexler, who actually shows people, what's the word? Not noisy patterns, ambiguous patterns. So in the visual system of humans, you can show people ambiguous motion, dots moving on a screen, totally ambiguous, right? And actually the perception of motion is your perception. The dots are not actually moving, they're appearing and disappearing.
Yeah.
And what's interesting is they're appearing and disappearing in the same way, but every individual has a different bias to report how they see that motion. Yeah. Now, if you test the same,
maybe somebody online could pull out the link to that. It's a classical experiment, the direction of the movements of the dots.
And if you now query the same individual multiple times, and people actually did in I don't know the year, there's a PNAS paper that I recently saw where they looked at about I forget how many people, with the exception of 3, over a 1-year period, with the exception of 3 individuals, an old classic paper, people coming back 3 months later reported exactly the same biases in. So not only is it in handedness, might also be in visual perception, just like in the flight. And so what we're trying to do is maybe develop a way in which we can do MRI while these people are looking at these screens and see if we see activities in the brain or
cool.
whatever. We're dreaming.
A little bit of a technical question. Are you concerned that variability in the DCN cell or axon number might instead reflect variability in the genetic line/marker used to label them?
Oh, I mean, yes. So we've gotten that question a million times. So basically, we use at least 3 different versions of this that are either the enhancer or simply a knock-in of the GAL4 into the locus, or the enhancer as a transgene in a different locus, and we can breed them to each other, and not only is the variation the same, but also the patterns in any given individual between different transgenes and different constructs are the same. So I think it's highly unlikely that the second thing that makes it unlikely is if when we change, as I told you, I didn't show you all the experiments we did to changing the wiring of the circuit, we can make it more symmetric or more asymmetric by our tools, and yet the behavior reflects what our tools predict. And of course, so it kind of makes it hard to think that our tools are biased and yet the behavior is regulated by our tools. I mean, that's, I think.
Okay. Maybe something about metaplasticity in a way. Is the extent of developmental noise and stochasticity within a population an involvable, inheritable trait?
I think one should ask Mark Kirchner about this. He has elaborate ideas on this. I don't know. Again, is the easy answer. But I would guess so, because I think that you have to constrain it, right? And I would suggest that at least some. So I'm going to go out on a limb here. I would suggest that at least some neurodevelopmental disorders. I think you and I, Denis, had a very brief Twitter thing. You tweeted a paper, and we discussed about the possibility that certain neurodevelopmental disorders may actually be mutations in genes that control excessive noise.
Yeah.
I mean, one of the simple things you can think about is if you have a chaperone that regulates the fact that the majority of proteins made in a cell should really fold pretty much normally. If you now reduce the activity of the chaperone, you can imagine that now you sort of expose all the possible range of random protein folding, which would reduce the amount of protein that folds properly, which would result in defects in development. So that would suggest that genes evolve that regulate noise, that control noise, right? So in a way, you evolve mechanisms that are stochastic in their nature, but you limit how noisy they can be, so that the curve looks like a normal distribution and doesn't sort of flatten out to something incompatible with life. That would be my guess.
Yeah. Okay, I'm scrolling through the questions. There's quite a few, but maybe we need to wrap up a little bit. You can also look at them. You can also handpick them yourself too if you click on Ask a Question. But let me see. I've sorted some out because they overlap with some that have already been answered too. There's quite a few questions on asymmetry, left-right asymmetry, but I think you elaborated quite a bit on that. Do you think that once the bundle is made, targeting of the medulla is dependent on guidance molecules or not?
Yeah, it almost certainly is. So you can reduce. I mean, we haven't done it in a temporally specific way, but if you go back to Srana et al. In 2006, which is one of the first papers where we started describing how the axons in the medulla behave. And the paper I quoted about crossing the midline and getting into the optic lobes. So it looks like a certain level of non-canonical Wnt activity and a certain level of frazzled activity, so netrin dependent, presumably, are important for the axons to either be attracted to the medulla to begin with, or to at least stay in it. And we haven't really looked at. So one could, look in a reduced netrin or reduced Wnt5 background as to how those bundles look. That's a great idea. Thank you for that idea. I don't know if Maeva's listening, but if she is, maybe that's a cool experiment to try.
Okay, I think we're reaching the end of our allotted time. Those of you who want to directly ask questions to Bassem, I'm sure he'll be happy to answer them at some time or another. So at this stage, what I really want to do is thank you again, Bassem. It was a lot of fun having you in. Very inspiring and interesting discussion.
How do I answer the remaining? There are questions that are remaining, and then there are questions within the chat box or something.
Well, in principle, there shouldn't be questions in the chat box.
Okay. So
I would suggest people that have pressing questions that have not been answered to send you a mail. Probably that's the simplest.
Okay,
And,
cool. Yeah.
And Bassem promises to answer by the time we're allowed to wander freely in the streets of our cities.
Yes. So is my email shared, or should I put it here, or what do you suggest?
Well, people can look it up, I guess. If it's urgent, they can look for corresponding authors. Before we say goodbye, I want to advertise next week's talk. So same time, same place, 5 p.m. In front of your computer on Thursday. It'll be Silvia Capello from the MPI in Munich, who will be telling us, her title is Following Neuronal Trajectories. And she'll be telling us a bit about disease, I guess, and also normal development, perhaps at the interface of what Bassem just alluded to in terms of pathological consequences of developmental processes. So looking forward to that. People, please do come. Until then, stay safe, all the best, and hope to see you soon. Bye-bye.
Thanks, Denis. Bye.